Hi guys, welcome to part 2 of lecture 25, which is coincidentally on November 25th. And this is installment 2, continuing on with calculating allelic frequencies and genotypic frequencies. I left you with an example that you were supposed to figure out. So here's the answer. The frequencies of each genotype are, again, just the number of individuals of that genotype divided by the total number of individuals. For alleles, it's slightly trickier, but I think you can do it, where it's the number of individuals for the M allele, the number of individuals with that homozygous genotype times 2 plus the number of heterozygotes divided by the total times 2, because each individual has 2 alleles. And one thing, then you would do the same for Q. One thing that is absolutely necessary when you are doing these types of calculations is to make sure that all of these add up to 1 and all of these, both of these, add up to 1. Just to double check yourself. All right, so hopefully that went well. All right, so now we're going to talk about Hardy-Weinberg, our favorite. And what Hardy-Weinberg does is it lets us have a kind of a baseline in an ideal population. This is what we would expect to happen. And what we would expect to happen is that the frequency of big A should equal P squared. The frequency of heterozygotes should equal 2PQ. And the frequency of recessive homozygotes, in this case, is Q squared. And like as with always, regardless of whether or not you're in Hardy-Weinberg equilibrium, P plus Q needs to equal 1. So that's a given. So this is really what Hardy-Weinberg is about. These are the genotypic frequencies that you would expect under Hardy-Weinberg, and then allelic frequencies are what they are. All right, so this is going to be true under Hardy-Weinberg equilibrium. If Hardy-Weinberg equilibrium, which I'm going to abbreviate HWE, and there are a number of assumptions of Hardy-Weinberg equilibrium. So we have Hardy-Weinberg equilibrium is true if first populations are large. Population, let's just say. We're just dealing with one at a time. Population is large. 2, there is random mating. And 3, there is no mutation, migration, or selection. Okay. So we'll call this natural selection just for funsies. Okay. So what this is really about, just to kind of place it in the context of where this lecture is going. The large population thing is really about, it means that when your population is large, it means that you have no genetic drift. And we will talk about what that means in a minute. If you have random mating, you also are not mating assortatively, which we'll also talk about that. And for mutation, migration, or natural selection, these are other evolutionary forces that can influence gene frequencies. So what happens under Hardy-Weinberg equilibrium, what this means is that populations, well, allele frequencies don't change from one generation to the next. And this means that the population is not evolving. Okay, because evolution by definition is changes in allele frequency. All right, so under Hardy-Weinberg equilibrium, we have no evolution. Allele frequencies are not changing, and this makes sense because we've gotten rid of we're basically saying we don't have any of these evolutionary forces. Genetic drift is also an evolutionary force, so we'll talk about that. Okay, what have I missed? So another thing about, oh, there's a clear question. All right, you can pause it here. Do your clicker, and it should make sense because all of the assumptions are here, and this is about what's not an assumption. Okay, you can pause that if you want to and look at that question. All right, so where do these come from? Hardy-Weinberg is an extension of Mendelian laws to a population. Extends Mendelian laws to a population level. So, just as under Mendelian law of independent segregation, I think, we have an equal probability of a big A or a little a joining up to create a zygote. So these probabilities come from the probability of big A times the probability of big A. So these are our rules of probability, our multiplication and addition. So here we have P, because P is the frequency, which is also the probability of that allele. We have P times P equals P squared, representing this genotypic frequency. We have Q times P equals PQ, and P times Q equals PQ. So this is 2PQ, which explains this guy. And then we have Q times Q, because Q is the frequency or the probability of little a. So the probability of getting two little a's in the same individual is Q times Q equals Q squared. So that's where these come from. And all right, so let's look at these assumptions a little more closely. How big is big enough for a population? Technically speaking, we're talking about infinitely large, which of course is not practical, but for all intensive purposes, Hardy-Weinberg is a-okay under most reasonably sized populations. It only, you get deviations from Hardy-Weinberg equilibrium only when populations get small. So then the next question is, how small is small enough? There isn't a threshold level of population shrinkage before Hardy-Weinberg equilibrium no longer applies. It depends on the species, and what this question is really about is there genetic drift. Genetic drift, which I'll talk about and explain what that means, but it's especially an issue, it's especially strong in small populations. So the answer is, how small is too small when genetic drift becomes a problem? Okay, random mating. So, what did I want to say about this? All right, so what this question is about is also, it's about sort of mating, but it's also about probabilities of gametes pairing together. And so just like we were assuming in Mendelian's law of independent segregation, I think that was his second law. I'm talking about his first one, which I think is assortment. So the first one meaning that you have an equal probability, which was, according to Mendel, 0.5 of getting a big A, and 0.5 of getting a little a. That's really applying it to, so Mendel was about the joining of gametes. In population genetics, we're about the joining of individuals. In Mendelian principles, we were talking about random joining of gametes. In population genetics, we're talking about random joining of individuals. And that's why we can have these predictions that the frequency of big A should be p squared. Okay? Okay. So in general, for the assumption that there's no mutation, migration, or natural selection, mutation at a population level is often negligible. And so population geneticists typically feel pretty comfortable with ignoring it. Often population geneticists are also really interested in migration or natural selection. And so what they can do is use Hardy-Weinberg Equilibrium as a baseline, as a default, as something to compare their population to say, hey, is our gene, our locus that we're interested in affected by migration? Is it under selection? And Hardy-Weinberg is kind of like a control for answering that question. So you can calculate genotypic frequencies in your population. You can calculate allele frequencies based on that. And then you can calculate what the genotypes would be under Hardy-Weinberg. We'll talk about all this. And then you can use a chi-square test to compare those. So, all right. So the main point of this particular sentence is that Hardy-Weinberg, the assumptions are often either okay or not okay, but when they're not okay, we're actually interested in whether they're not okay. And so we can use Hardy-Weinberg as a point of comparison with real populations to determine whether or not they're behaving in an ideal population way, and if they're not, why not? Okay, Hardy-Weinberg also applies to single loci, to one locus at a time. And so this particular case, just the A locus. If you want to consider population genomics, so what are loci across the entire genome doing within a population, then you can apply Hardy-Weinberg principles to each locus one at a time. And what you'll find is that some loci are under Hardy-Weinberg equilibrium and other loci are not. And so that's a research question that people can also investigate in population genetics. Okay. So, Hardy-Weinberg means that populations are not evolving. And it also means that we can use Hardy-Weinberg law to predict allele. Let's see. We can predict genotype frequencies from allele frequencies. We can then compare that with the actual genotype frequencies in the population. Okay, so let me rephrase this. So we can